The Black and Biophysics Symposium brings together researchers from across the field to highlight emerging discoveries and the scientists behind them. We are joined by Heather Pinkett, whose work explores how cells transport molecules across membranes, processes central to nutrition, infection, and cellular survival. Thank you for being here.
Thanks for having me.
So Heather, your work, it really focuses on ABC transporters. What are those proteins actually doing inside of our cells?
Yeah, so the responsibility of an ABC transporter is to transport substrates across a membrane. And so you have importers that are bringing in nutrients into the cell, especially in bacteria and archaea. And so exporters are responsible for transporting out toxins, but they can also change the composition of lipids in the lipid membrane. And so you have these two proteins that they're responsible for both bringing in nutrients and taking out toxins.
Very nice. You often also refer to them as molecular turnstiles. What have you learned from the energy of the ATP and how it drives the motion?
Yeah, so through both biophysical studies and structural studies, we know that an ABC transporter consists of like two components. There's a transmembrane domain that's located in the membrane, and then a nucleotide binding domain. So the ABC in ABC transporter stands for ATP binding cassette. And so ATP binds to this nucleotide binding domain, and when it binds across the interface. And so you can imagine when it binds across the interface, it brings those nucleotide binding domains together. Once they're brought together, it kind of communicates to the rest of the transmembrane domain, and that transmembrane domain opens up, much like a turnstile. So you can imagine you're going through a turnstile, you put in your coin and pay your fare, and then you get transported into or across that membrane. And it's the same thing with an ABC transporter. That ATP binding and hydrolysis, that energy from it, opens and closes those gates to allow nutrients in or toxins out.
So for the everyday person who's not as familiar as the science, but they just know bacterial infections are very serious, and when it comes to these transporters and how it relates to bacterial infections and the drug resistance, what do we know now? I mean, what does this tell us?
Yeah, no, that's a really good question. So for many ABC transporters, we know they're virulence factors, which means if they're stopped, mutated, inhibited in any way, it makes the bacteria more susceptible to killing. And so you can imagine when we're solving these atomic structures of ABC transporters, we have an understanding of how they recognize their nutrient. And if we know how they recognize their nutrient, it's basically a playbook for us to design these inhibitors.
From the research that you have presently, where do you see the future going here? And is there a timeline?
Yeah, so for a lot of the research that we're interested in, we really want to know how in bacterial systems, how these particular transporters are able to bind to antimicrobial peptides. And so what we're looking to do is inhibit them from binding to antimicrobial peptides that are produced by our own host innate immune response. And so if we can increase the concentration of those antimicrobial peptides by turning off a specific transporter, that means our body can clear an infection in a way that doesn't require us to use a number of different antibiotics, and we can slowly stop the multidrug resistance epidemic.
Which would be absolutely profound and breaking, I think, medically for everyone. How does it feel with the symposium coming up to just be recognized and to share this message in your research with everyone?
Yeah, it's absolutely wonderful to be back at the Biophysical Society meeting. This is a meeting I've been going to for many years since grad school. In addition to that, to be in the Black and Biophysics session holds a special place for me, just because Black and Biophysics is a wonderful organization. It's a grassroots organization created by graduate students and postdocs who really wanted to build community and show undergrads that, yes, there are Black biophysicists out there. And so to have this collaboration with biophysics and the Biophysical Society and Black in Biophysics is really important. So I'm really excited to give a talk in that session.
Wonderful. Well, the work that you do is really important. So thank you for joining us and being here today.
Thanks for having me.